Hi, my name is Ashley, and today I'll be talking about the CRISPR genetic system. First off, what is CRISPR? Identified first in E. coli. It stands for Clustered Regularly Interspaced Short Palindromic Repeats. Researchers have stated that it it was stated to revolutionize medicine in the way we fight diseases and to cure cancer and amongst other things. I know that it's a mouthful. It's a mouthful, so I am going to start by breaking each letter down for you. Starting with the SPR. Now, the SPR, starting with the S stands for short. It is a short segment of DNA. The length of letters is no shorter than 20, no longer than 40. So you have your short strands of DNA. Palindromic, it is breaking down the word. The omics means all of something, which palindrome is it reads all of the short strands of DNA from left to right. That's what palindromic means. Now you have your repeats, and repeat is just like the word says. It's like if you are in your car and you have your CD player on repeat, you're going to keep repeating the same song over and over and over again. In this case, you're going to continue to repeat the DNA, the short DNA strands, over and over and over again. They are identical. Next, we'll be talking about the CRI of the CRISP word. C, it stands for your cluster. So, of course, as cluster means, it's a cluster of your DNA together. Regularly interspaced, that is a word combined, is that inside of the spaces, you'll have spaces in between the short strands of this DNA. The regularly interspaced spaces, it distributes space in the middle of each short DNA, which you have distributed here. Now, the spacer DNA, which is, these are the spacers. Now, those spacers aren't just space. I should clarify that. Inside those spacers will be DNA, but you may think it will be the identical or match up with the already repeated identical DNAs here. No, in the spacers you have very unique DNA inputted into them. The spacer DNA is not identical to the original DNA, which right here, if I don't know if you can see, but in the spacers, that one's blue, that one's orange. Each segment of the spacers is going to be unique. Researchers found out that the spacer DNA matched up with viral DNA. That's different viruses in our society. Okay. Now, a number of genes associated with CRISPR have CRISPR associated or something called Cas, C-A-S, genes. Now, Cas stands for, which I don't know if you can see it, but here's your Cas. And in the Cas, they did a yellow highlighter that shows you like the three Cas right there. But basically, Cas is a gene that produces Cas proteins. Now, you have two different types of Cas. You have your helicase that unwinds the DNA, and you have, well, not two different types of Cas, but the Cas proteins are, it is helicase, which unwinds the DNA, and you have your nuclease, which cuts the DNA. So you have one that is zipping up, and you have your nuclease that is cutting down like scissors. So here is basically, I can't get the swirl of the DNA, so I just drew it a straight line, but these two right here are spacers inside of your repeated regularly DNA. And in the spacers, they have their unique types of DNA entered in. So this blue one is unique, and this pink one is also unique. All different. Now, got it upside down. Now let's break it down with this systems. So right over here, let's go back to that. We have our E. coli. This blue line right here is your cellular membrane. Then right down here you have your genome strand, and over here with the colors I have your Crispr, and over here you have your Cas genes, which you really can't see on here, but they're highlighted in yellow. But the process goes like this. So your E. coli, which is your virus, your bacteria, comes and connects to the cellular membrane. Here's the bug right here at the cellular membrane. Okay, so at the cellular membrane, it injects its viral DNA inside of the cellular membrane. Now, a human without a immune system, this viral DNA will begin to replicate and eventually kill the cell due to its replication of its virus inside of the cell. But with the CRISPR genetic system, what happens is when the E. coli injects its DNA inside the cell membrane, the Cas translate and transcribe a protein, which is located right here. And the Crispr, which it transcribed into Crispr RNA, and it injects it inside the protein like it's, my analogy is I call this protein a car, and you have your kryptonite inside of the car. It fits snugged right inside of there. So once this DNA, and as you see, it gives you the gene that's identical to what DNA bacteria that was injected in. So this DNA strand will come down, get eaten up by this gene and destroyed before it even starts replicating inside of the genomic system. That is, to me, that was very cool. But what if that unique strand is not already inside of the genomic DNA? Well, that's when this example over here come into play. The E. coli will inject a unknown virus. It A unknown virus